So, have you ever wondered what really goes on inside an IVF lab? I mean, behind those closed doors. Well, today we're going to pull back the curtain. We're going to take a deep dive into that incredible journey from oocyte retrieval all the way to embryo culture and really get into the science that makes it all happen.
All right, so here's our game plan. We're going to start where it all begins, way before the patient even gets to the clinic, with all that meticulous lab prep. Then we'll follow the process right through to the end, selecting the very best embryo for transfer. Let's get into it.
Okay, so first things first. A successful IVF cycle, it doesn't actually kick off in the operating room. Nope, it starts right here in the lab. And not just a few hours before, but often a full day before the retrieval. This is where the foundation for everything, and I mean everything, gets laid. And you better believe they have a checklist. Think of it like a pre-flight checklist for a pilot. It is absolutely non-negotiable. Every single item has to be checked off. We're talking viral screening results, all the consent forms signed and double-checked, and maybe most critically, labeling every single dish. There is just zero room for error. This is ground zero for patient safety.
So once all that administrative stuff is squared away, the focus pivots to something purely scientific, creating the perfect environment. The whole goal here is to build a little home away from home for the embryo, one that mimics the conditions inside the human body as perfectly as we can.
Now, if there's one single factor that is the most critical to control in this environment, it's something called osmolality. You can kind of think of it as the saltiness or the concentration of the liquid the embryos live in. And let me tell you, embryos are incredibly sensitive to this. I mean, even a tiny, tiny shift can throw the fluid balance inside their cells completely out of whack. And that can be disastrous for their survival.
So the big question is, how do labs keep this balance so perfectly stable? Well, the number one enemy, the big villain in this story, is evaporation, right? A little bit of the water evaporates, and suddenly the medium gets more concentrated. The entire lab workflow is basically a battle plan designed to fight evaporation at every single turn. And here's how they do it. Embryologists have this really strict protocol. They work super fast, they use cold media straight from the fridge, and they use larger droplets. And then, this is the key, they immediately cover all those little droplets with a protective layer of mineral oil. That oil acts just like a blanket, It locks in the moisture and keeps that crucial osmolality right where it needs to be. Perfectly stable. And speaking of keeping things stable, check this out. This is where things are heading. We're starting to move beyond just these static little dishes of media. We're looking at things like microfluidic devices. The idea is to create a dynamic flow of nutrients, something that better mimics the embryo's actual journey down the fallopian tube. It's like upgrading from a still, quiet pond to a gently flowing stream. It's just so much more natural.
Okay, so the environment is perfectly prepped and waiting. Now we're ready for the main event, the retrieval procedure itself. The test tubes of follicular fluid arrive hot off the press, straight into the lab, and for the embryologist, the hunt is on. Time to get on the microscope.
Peering down the microscope, the embryologist is scanning through all that follicular fluid, and what they're looking for is this fluffy, kind of shimmering clump of cells. That's it. That's the cumulus oocyte complex, or COC. And tucked away, right in the middle of that little cloud, is the prize, the oocyte itself. Now, especially for a procedure like ICSI, that's where they inject a single sperm right into the egg, they have to clean off all those surrounding cumulus cells. And this gives them their first real, naked look at the oocyte. And you know what? They're not all perfect. Some of them have unusual features. We call it dysmorphism. Things like little bubbles, called vacuoles, or grainy spots. These are all carefully noted because, they can sometimes be a sign of lower developmental potential. All right, so the oocytes have been inseminated, whether that's through standard IVF or the ICSI procedure, and now the lab waits. It's this quiet period of anticipation. Then, the next morning, usually about 16 to 18 hours later, comes the moment of truth. It's the first huge milestone, the fertilization check. And this is the money shot. This is what everyone is holding their breath to see. A normally fertilized egg, which we now call a zygote, should have exactly 2 little circles inside. These are the pronuclei. One's from the egg, one's from the sperm. If you see 3, or if you see none at all, well, that's not good news. It means the zygote isn't viable and can't be used. With modern time-lapse technology, we get a front-row seat to this whole process, and it's like a perfectly timed ballet. The pronuclei show up at around the 6-hour mark. By hour 9, they've snuggled up next to each other. And then, at about 25 hours, they fuse together. And that happens just moments before the embryo's very first cell division. It's just incredible to watch. So over the next couple of days, the embryos just keep dividing. This is called the cleavage stage. And during this time, we've got to keep an eye on them and assess their quality, right? We need to figure out which ones are the top contenders for transfer. And honestly, even with all our technology, this process is still as much an art as it is a science. And this brings us to a really fundamental truth in embryology, one that can be kind of counterintuitive. What an embryo looks like, its morphology, it's just not the whole story. This quote here says it all. You can have the most beautiful, textbook-perfect embryo that's actually chromosomally abnormal. And at the same time, one that looks a little poorer in quality might be perfectly healthy. Bottom line, looks aren't everything. So when an embryologist is grading, they're kind of like a judge at a competition, looking at three main things. First, the number of cells. We call those blastomeres. Second, how even and symmetrical those cells are. And third, the amount of fragmentation. That's just little bits that have kind of broken off. And you can really see the difference here, right? Look at the uniform high-grade embryo on the left compared to the one with more fragmentation. Now, for a lot of cases, the journey in the lab doesn't actually stop at day 3. We can let the embryos continue to grow for a few more days, out to day 5 or even day 6, until they reach what's called the blastocyst stage. And this extended culture, this waiting game, it gives us another really powerful selection tool. And the thinking behind growing them out to day 5 is really solid. It's basically a marathon, a test of developmental toughness. See, around day 3, a huge thing happens. The embryo's own genes have to switch on and take over. It's a massive hurdle. And only the strongest, most robust embryos can make it through that transition to become a blastocyst. In a way, they kind of select themselves. So when we grade a blastocyst, the first thing we look at is its expansion. It all starts with this little fluid-filled cavity, the blastocele, that forms inside. And then that cavity just gets bigger and bigger until the whole embryo swells up, which thins out its outer shell. And then finally, it hatches right out of that shell, all ready for implantation. Okay, besides expansion, we also have to grade the 2 different types of cells. First, there's the inner cell mass, or ICM. This is the good stuff. This is what's going to become the fetus. We grade it based on how many cells there are and how tightly packed they look. Then you've got the trophectoderm. That's the outer layer, and it's destined to become the placenta. Again, we're looking for lots of cells that form a nice, cohesive layer. For both, an A grade is what you're hoping to see. And here's a perfect example. Take a look at this gorgeous, fully formed blastocyst. You see that big, clear bubble? That's the blastocele, all filled with fluid. And that dense little clump of cells tucked to one side, that's the inner cell mass. And then you have that perfect, tidy layer of cells all around the outside. That's the trophectoderm, just textbook. But just when you think you've got it all figured out, we come back to reality. This slide right here is such a powerful reminder of those limitations we talked about. This particular embryo, it wouldn't get a perfect score. You might grade it as lesser quality, and yet it resulted in a healthy, live birth. It just goes to show you the massive challenge we still face when we rely only on what our eyes can see. And that really leaves us with the biggest question in the whole field, doesn't it? If we want to keep improving outcomes, we have to find ways to move past the subjective art of just looking at an embryo. The future has got to be in finding objective, measurable markers, things like analyzing an embryo's metabolism, or even better, its genetics. That's how we're really going to find that one single embryo that truly has the best possible chance.